these are very interesting because they act like normal tissue. Now again, you've got to think in terms of long ages. If these are 67 million years old, they shouldn't behave like regular tissue. I shouldn't be able to thin section them, but I can. I shouldn't be able to stain them, but I can, and they take up the stain. So they're behaving exactly like my tissues are, yet they're supposed to be so old. So it's really kind of an oxymoron. Another charge was these are simply pieces of fungus that were growing in the bone. Now you can do a test. You can do an optical test for fungi or fungi, whatever pronunciation you use. And if you stain them, you'll see crossbars in them. But you see no crossbars in these cells. So these are not a fungus. All right? These are actual cells. Now other scientists have found actual molecules inside of these cells. They've done very sophisticated work, and they've actually found the individual molecules that are still there. Someone mentioned in the earlier talk that DNA, you know what DNA is, right? That's your genetic code. That has a half-life of 500 years. Okay, what does that mean? That means if you have a pound of DNA, in 500 years, half of it will be gone. In another 500 years, half of that will be gone, and so forth. So DNA doesn't last very long. But they're doing tests, and we've done tests, and we're showing that the DNA, those molecules are actually still there. So this is now accepted science by those who are willing to accept it. Another charge is that they've turned to rock. They've mineralized. And I've mentioned to you that none of these bones are mineralized. They're still bone. I can still dissolve them. Maybe when you were young, you took a chicken bone, and you put it in a jar full of acetic acid, right? Vinegar. Anybody ever done that before? Or you should try it. Soak that chicken bone in vinegar for about a month and take it out, and it will be floppy. Why? Because you take all the bone mineral out. The bone mineral gives it stiffness. So that's what we're doing. We're soaking these in a weak acid, and they just dissolve, and all the soft tissue falls out on the Petri dish, and then we can photograph it. So they're saying they're mineralized. Well, if they're mineralized, they would not dissolve. Rock does not dissolve in acetic acid, right? So here's a picture of a cell, and this is called bright field, so it's just bright. But if we look at it in polarized light, we can test for mineralization because minerals glow in polarized light. So here's the same cell in polarized light, and you can see the difference. Is there a lot of mineralization in that cell? Hardly. Okay? In fact, I searched and searched and searched to find mineralized cells in the bones, and I couldn't find them. Here's one in bright field. There it is in polarized light. Is it heavily mineralized? Doesn't look like it. So searching, I found one, and here's one that is heavily mineralized. This is all calcite. And here's another one that's partially mineralized. So these are still bone and soft tissue. They have not been changed. This is impossible. It's, according to everything I know as a scientist, this shouldn't be there. But it's there, and it's in every bone that we've tested. So osteocytes are a really great tool to look at and to be able to understand what was going on while they were there. Now, there's another technique you can use where you hit your cells, you hit the soft tissue with, say, blue light, just one wavelength of light, and it glows. It glows another color. Remember when Moses went up on the mountain and he came back down? What did they make him put on? He had to put on a veil. You remember that? Why? Why did Moses have to wear a veil? His face was glowing because he'd been in God's presence. So the light of God got his tissues to fluoresce. And it was so scary to the Israelites, they made him put a veil on, right? They didn't want to see that. But tissue does glow. And I think this has implications for our glorified bodies. You know, what are we going to be like in heaven? I think when God's light is going to impinge upon us, we're going to glow with his light. So let's hit this cell with blue light and see what happens. Now that's impossible. Why? There should be no proteins in this cell at all. Those are molecules. Molecules don't last, right? But it's glowing. Inside we see proteins glowing. Let's look at another one. All right, here's one in bright field. Nice cell. You can see the filopodia sticking out, right? Sometimes you can see the nucleus. You can still see a nucleus in these cells. There it is in fluorescence. And it's glowing. See all the little points it's glowing in here? Those are all proteins that are still in the cell. Now, Dr. Mary Schweitzer from North Carolina State University is world famous for this work. And she did a whole slew of papers proving over and over and over that this is actin, this is tubulin, proteins that are part of the cell that shouldn't be there. And she proved conclusively that they are there. Most of the scientific community doesn't want to talk about it. A small part of the scientific community acknowledges, okay, yes, these dinosaur tissues are real. But most of them say, that doesn't exist, that's a myth. So they even refuse to accept the published papers from one of their own scientists.